Reproduction. Now, as I've indicated before in class in various different ways, when we talk about reproduction, that is the process of making more. So we will talk about a sexual process. We're talking about mixing of genes that is incorporated into reproductive processes. But of course, they are two different components. A couple things to remember is that we'll talk about various different components, particularly in the interphase, the space between the cell cycle or cycles of the cell, and talk about how those particular components really how those components relate to some of the stuff we'll talk about in Unit 3 when we're talking about DNA replication and whatnot. So it'll kind of all come together, but we're kind of giving you the basic pieces, and then we're going to fry up a delicious cassoulet of information, bringing all these different pieces together. So all cells come from cells. We talked about that a little bit already with the cell lecture, basically cell theory. And of course, making new cells and often making new organisms all, of course, require that process of cellular reproduction. We start from a simple single cell, a fertilized zygote, and from that we go through trillions of cell cycles, and of course continue to do so throughout our entire life. Because of course, while some cells will kind of not really reproduce as often, there are large components of our skin and a variety of other tissues within our systems that do require new cell material. And so we will see that there are those particular organs, are those particular systems that require constant mitosis. And then, of course, towards the end of this component, we'll talk about cancer being a situation where the cell cycle has gone out of control. So we'll try to see what we can do to understand about that related to this important thing. So asexual reproduction, a reproductive process, so you're making more, but it's asexual. It does not involve any form of genetic recombination, does not require, it is a component, but it doesn't introduce variety, so you don't have new gene combinations and things like that. And of course, we will see that process, and in the process of sexual reproduction, when we talk about Chapter 9, the meiosis, you will of course see that it goes through two cycles of sorts, and those cycles themselves do show a reproductive component. You go from 1 cell to 2 to 4. So the two main processes in the cell cycle or in asexual reproduction is cell growth, where during this process it grows, and part of that is duplication of its contents. The duplication of DNA, which, as I said, we'll talk about in the next couple of weeks after we will do some stuff with our good friend Mendel and those sorts of genetic questions. And again, all of these pieces kind of reinforce each other in different ways. So we want to kind of approach this question at so many different angles. So there is that process of cell growth where there's duplication of contents. You want to make sure each of the daughter cells get the material important to those cells so that they'll be identical to each other and identical to the parent. The other component of asexual reproduction is, of course, cell division, where in fact the parent cell will divide and you actually have that that process of separating the two pieces. And so obviously there is a continuous cycle. They always are informing each other, and all of them have checkpoints and are regulated within the cell in a variety of different ways. So you definitely have things controlling those steps. And again, this is kind of giving a foreshadowing to what happens when those regulations are not well done or are no longer functional, and how that can lead to, as I said before, things like cancer. So the structure of DNA, we mentioned this briefly when you looked at cells, and when we looked at cells in general. When we had a nucleus with a nucleus with a nuclear membrane, the membrane, of course, identical to the membranes of the outer cell and to all the membranes found throughout the cell, with things like endoplasmic reticulums and Golgi and vacuoles and the like, as well as the numerous layers around the mitochondria and chloroplasts, etc. But one of the things that we saw was the DNA was kind of in a very loose component, and that is called chromatin. Of course, you perhaps talk a lot more about chromosomes, the some being a body. And what you can see is that the chromatin will condense around these proteins called histones and wrap around these histones to create larger and larger. It's sort of a very thin rope that gets kind of created into a zigzaggy larger rope, which then loops, and so on and so forth. Ultimately, you end up with, when the genetic material is fully condensed in the form of chromosomes, you can see the entire genetic material all condensed into, all that material within the nucleus is condensed into multiple chromosomes, 2 or more chromosomes within the cell. As we'll talk about, of course, most humans have 46 chromosomes. We'll talk about those. Obviously, that information and material kind of splashes over onto sex-linked traits in Mendelian genetics, and of course, the process of DNA replication. So, like I said, these are all really important connected concepts. So when you're looking at a chromosome, when you're looking at the actual sort of material, you can see that in this case, there are actually 2 pieces, or 2 sister chromatids, which themselves are connected by a structure known as a centromere, and the chromosome is made up of these 2 sister chromatids who are identical to each other. This genetic piece of information and this piece of genetic information is identical, and that is because it's gone through the process of duplication, which we'll talk about, like I said, in the next few weeks. So when during a process where the cell cycle isn't very active, during times of growth, the DNA is very loosely arranged within the nucleus, and then as it goes into a cell cycle, it is condensed around these histone proteins, et cetera, until you have these actual chromosomes found within the cell. Now, something to think about, again, for those of you who've perhaps had a little bit more education behind transcription and translation, you can think about how, if the genetic material was all packaged up tightly with all these networks and loops and proteins and all this sort of thing all together, you really would have a hard time saying, okay, this little stranded gene, I'd like to transcribe it and see what kind of proteins we create from it. That's just not going to happen. So during normal growth of a cell, the genetic material is very loose in that in that chromatin. And as a result, it will allow it to be a lot more easily utilized as we will talk about with the different components of DNA. Okay, so there are basically 3 steps of the cell cycle. There is interphase, mitosis, and cytokinesis. So interphase is a period of growth, and we'll talk about some of the different things that go on in interphase. Mitosis is the prophase, metaphase, anaphase, telophase that will go through the period of division, particularly of the nucleus, because this is division. What we're focusing on is division of the genetic material. And then after the genetic material is divided, we'll also have cytokinesis, the division of the non-nuclear contents or the division of the rest of the cell. So, as I mentioned, you've got a very large interphase. As you'll see when you're looking at under the microscope, we'll look at a variety of different cells going through cell cycles, and you'll be asked to kind of look at some of the different structures and see, when you look at the cells, what stages they're in. And what you'll see is this cartoon here isn't just an indication of sort of the process of the cell cycle, but it also gives the idea of how much time each of these different sections are. And what you can see here is that interphase is by far the most predominant stage within a particular cell cycle, and the other stages are considerably shorter. And within the mitosis process, this is going to be, you'll see different stages having different components. You can see the anaphase is relatively a small piece of this little pie. So when we're talking about interphase, this is just basically a majority of the time when it's doing its functions, all the sorts of things that cells do. Again, it depends upon what type of cell you're looking at. There are some cells like muscle cells there are some cells like muscle cells and neurons that don't necessarily that kind of spend almost their entire lives through interphase, and we kind of think about why that is. So what happens during interphase? There are 3 different components. There is the G1 stage, the S stage, and then the G2 stage. Those are the 3 components of interphase. And here you have a lot of growth. As organelles double, you have multiple, creation of new material within the cell, so that ultimately when the cell divides, both of the daughter cells will get some nice juicy organelles. You can see here that, like I said before, something like your neurons might kind of perpetually be in a G sort of sub-zero cycle of it, that there isn't actually any process of cell cycle after those particular cells have developed in the human body. After the G1, after this area of growth, you have this S, and this S is where DNA replication occurs. This is where the DNA makes a copy of itself, so that when it goes into mitosis, when it goes through this process of nuclear division and the material, the DNA in the nucleus dividing, you have enough to make 2. Boom, boom, 2. After the S stage, of course, so that's chromosome duplication. And then after the S stage, you of course have the G2 that will then prepare it for this big process of mitosis. Okay, so we can go more DNA. So the organelles, it accumulates. Should it in fact divide or not? And then, as I mentioned, those arrested in the G1 cycle or the GO cycle, these are not cells that are going to divide. Each chromosome, we talked about the chromosomes, when the chromatin all kind of got to a point where it's brought together and condensed. The condensing of these chromosomes are in fact due to you have the two sister chromatids identical to each other in that process. And again, before you do this, you're really not going to have you're not going to condense the chromosomes when you're copying something. If you wanted to copy like a piece of paper and the writing on it, well, you'd have a nice open flat piece of paper where you could look at all the different letters on that piece of paper. If, on the other hand, you condense that into a teeny ball, if someone gave you a small ball of paper with writing on it and said, hey, could you copy this? You might be like, I kind of need to unfold this to do this right. And that's true. Same thing with your chromatin. Same thing with your genetic material, loosen the nucleus until condensing as a part of the cell cycle. So this DNA replication, we'll kind of give you the short version first. The result is this copying of DNA. And when you look at a chromosome, of course, you have the centromere here, you have the two duplicated chromosomes, you have the two sister chromatids identical to each other. This one we'll see is a single chromatid, and we'll see where that goes, but it goes through DNA replication, creating two that are identical to each other, so that when you divide the cell, as we'll see, you will divide this chromosome in half, so both of the daughter cells, both of the resulting cells, will have the same exact genetic information. The chromosomes are composed of sister chromatin and centromere, and the two sister chromatids are identical to each other. All right. Finally, the G2 stage before getting into mitosis. It synthesizes the proteins, everything you need to put together this sort of thing, everything you need to kind of go into this process, into this cycle, and that then sends it into the process of mitosis itself. So when you look at reproduction, making more, asexually, no genetic combination or variation, the two daughter nuclei produced by mitosis are identical to each other and to the parent nucleus. Another important thing is that every animal has an even number of chromosomes. Each parent contributes half of the chromosomes to the new individual. And we'll see that's the case, of course, for meiosis and sexual processes, but that is, of course, also the case in your own cells, in your cells that are dividing by mitosis, because you have this even number of chromosomes, and half of those chromosomes are your biological father, half are your biological mother, and you go from there. And again, these multiples, this whole information really does connect to things that relate to that monk hanging out with bees back in the late 19th century, Gregor Mendel. So mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. Sounds hauntingly familiar. Plant and animal cells, though, have a couple of main significant differences. And we touched upon this a little bit when we did chapter 4, talking about parts of the plant and animal cell. There are centrioles in the plant, remember that's a pretty big component of the cytoskeleton versus the animal, which does have centrosomes composed of centrioles. And so they have, in a way, the same part, but the structure of their skeleton is dramatically different. Of course, there are other differences between plant and animal mitosis that we'll talk about shortly. But the centrosome itself is a location, or it's a structure that will organize the overall microtubule structure that is utilized in mitosis. And we'll talk about a spindle. This is a network, or a web-like network of microtubules within the cell, within the nuclear area, that will each attach to the centrosome and to the actual duplicated sister chromatids. The spindle itself, again, part of the cytoskeleton, as I mentioned already, this is the component that will break the centromere and will separate the two daughter chromatids and pull them apart. So, in terms of a combination of sort of some photographs, some micrographs, and some cartoons, you can kind of see some of the early stages here. Within interphase, you can see the centrosome made up of centrioles in the animal cell will start to organize and start to create spindles with a centrosome on either side. While that's happening, you also see the nuclear membrane is beginning to disappear. And so in prophase, the chromosomes are starting to condense. You have chromatin, that loose genetic material that is condensing to form chromosomes. It has gone through DNA replication if it's gone through the S stage, which it would have if it goes on to G2 and into mitosis, so that when those chromosomes do constrict or contract, they will form, the sort of with the two sister, the chromosome itself will be a full chromosome with two sister chromatids. And here you can see the genetic material is starting to condense. And then in this process as well, the centrosomes will migrate along either side of the cell, or either side of the cell, but really around either side of the nucleus. So as the nuclear membrane disappears, this spindle will begin to attach to the centromeres and again have its function to split apart the chromosomes to the sister chromatids. And these are just starting that process to have the spindle attached to these chromosomes. So that's prophase. Metaphase is indicated by the time when you actually have all of the genetic material, all of the chromosomes, the double-stranded chromosomes, are in fact lined up. So for humans, 46 of these in a row, all in a row, 23 from mama, 23 from papa, all in a row. The mama and papa is indicated by the red and the blue. I know of a couple ferns and maybe a couple flies that only have 4 chromosomes, but it's a lot easier to just look at 4 than to try to, stuff this with 46 odd chromosomes. So metaphase is very much indicated by all of the genetic material lined up. So you'll see the genetic material lined up when you're looking under a scope, and you'll see that this spindle is very much fully formed. and you have from the centrosome what becomes the aster that actually will help bring this back. So when you're looking at a cell and it has all the genetic material lined up and it has a nice spindle around it, you know it's in metaphase. Now the spindle will begin to contract. And look how pretty those little spindles are, contracting, pulling either side. And what they've done is they've split the genetic material in half so that you have a situation where both will get, you know, this piece, this little chunk right here and this little chunk right here is each the same, identical to each other, and each of them will get that whole process. Now with telophase and cytokinesis, what you then have is the beginnings of the breakdown of the spindle, the pinching off of the edge of the cell. So you go from one cell, one bigger cell, to two cells, identical to each other, identical to the parent cell. So that is that process of cytokinesis. You also, during that time, will begin to have this genetic material will begin to loosen up a little bit, go back to more of a chromatin sort of situation, and you will have the reformation of the nuclear envelope. So, prophase, and metaphase, anaphase, telophase. Very important events in this process. So the cytokinesis process is, of course, the process where one cell becomes two. The nuclear replication and division all happened in mitosis, but it is not until you have the separation of the genetic material, not until you have the separation of the genetic material and the separation of the two of the cell into two separate cells that you really have the full cell cycle going to two cells. There are, in fact, some weird things. There are some weird organisms. There's like some seaweed where it goes through nuclear division, but it doesn't go through cytokinesis. So you have these huge, enormous cells with dozens or hundreds or more nuclei in it. It's pretty amazing stuff, but we of course like to focus on not just animals, but on the stuff that generally happens, the rules rather than the exceptions. But sometimes the exceptions are pretty cool. And if you find one of these seaweeds on the shoreline, they're crunchy and delicious. Again, another issue. So what happens in the process of cytokinesis? Yeah, and actually muscle cells are a multinucleated cell. If you've ever looked at those cells under a microscope in a histology class or in one of those, you can really see that process. So animal cells, what actually happens? You have what's called sort of this invagination known as the cleavage furrow. So you have a pinching inward, but you also have this structure of like a contractile ring that kind of pinches around it, like a sphincter muscle kind of pinches around it, pinching off either side. I guess titling like a belt is perhaps a less vulgar way to put it, but yes, essentially the same process is happening. Now in plants, what's interesting is, of course, number one, you do not have their cell walls are very structured, and cell walls have, as you maybe remember, a ton of structural polysaccharides, a lot of fiber, a lot of really structural material. So instead of creating, instead of pinching off like animal cells can do, essentially there's what's called a cell plate, which forms in between the two cells. And this cell plate here kind of is a situation where it will create more membrane on either side, and then external to that membrane will be the formation of cell wall, because they do have a plasma membrane just like any other cell, but they do of course have the cell wall that makes them very again structured and whatnot. So they can't furrow, so instead they create this structure of membrane and wall separating the two daughter cells. These are created in vesicles, so you could certainly have things occur in the Golgi, which then sends these things to the cell plate. And then you again go through the process of manufacturing new cell wall between those two, which as I've indicated, is a hobby of mine, I guess you would say. Now, between the cells, you, of course, as we said before, we have the pectin. We talked about all that good stuff. So we've got some points to ponder. Why is there a difference between plant and animal asexual reproduction? Well, of course, you have a situation where you can't really, you can't have a cleavage furrow. You can't furrow a plant cell, but you can an animal cell. And the significance of having two of each chromosome, one from each parent, is of course within the biological species, and when you were created, you of course were the output of a sexual reproductive process. It was reproduction because you were a new individual, and so your parents sort of went from two parents to three, two people to three people, if you will. And of course, you are different from each other, and you get all of the good stuff. So, as I've kind of hinted at before, the cell cycle is extremely important. We need to control that because it assures that things happen when they should. All of these different components don't just sort of happen willy-nilly. And so there are actually checkpoints that the cell will then go through. Sorry about that. There are checkpoints the cell will go through that will determine whether it can go into the next stage of the process. A G1 checkpoint to indicate whether it's ready for the S phase, ready for DNA replication. A G2 checkpoint to ensure that that the DNA replication in the S phase has happened successfully. And then after the G2, there would be a mitotic stage checkpoint where it determines whether it's okay to go into the full mitotic process, separating the two, or separating the genetic information within the nucleus of the cell. So in this case, the G1 you can see occurs here. You have a situation where the G, if it doesn't work out, it will go into the G0 cycle if it's not ready until it can prepare. Or again, this G0 might be sort of perpetual. Apoptosis. What does that mean? Apoptosis is when you have cell death. Apoptosis is when you will have the destruction of cells or cellular material within the actual process of the cell cycle. So this is a situation where the cell might not really want to go on. The cell cycle would be impossible, and there really is nothing that could be done with the cell that was going through this process. And so this is sort of a form of selective cell death with apoptosis. It is something that happens by design. It's not always just something that is a way of kind of checking or a part of a checkpoint. For instance, sometimes or in the development, say, of the hand, there are certain cells between the fingers that go through this apoptosis. And just because this is a process that is scheduled or supposed to happen, it doesn't always. So you do end up occasionally with newborns with some webbing on their feet, between their toes or on their hands, and a surgeon will deal with it and they move on with their life. They're not better at swimming or anything like that. It's just sort of a part of life, and the apoptosis there didn't happen. But it is, again, an important component. Your G2 checkpoint ensures that the DNA is replicated. And then, of course, the mitotic stage, this occurs actually between metaphase and anaphase to ensure that once you have the spindle lined up, all of the chromosomes are where they should be, attached along the center of the cell, as you see in metaphase, ready to be split apart. So there are, in fact, these important checkpoints. Now, what causes these checkpoints to turn on and turn off? What causes these cells to suddenly say, hey, I'd like to make more? That sort of thing. Well, there are a lot of different growth signals that we can find. One good example is epidermal growth factor. This is a factor found within the body, which perhaps results in the formation of cell cycles near an injury. Of course, this is done more than perhaps normal because there is an injury. And of course, this can lead to things like scarring. Estrogen itself actually stimulates division of cells within the uterus to prepare for egg implantation as part of the cycle in the uterus. Not every cycle or not every signal says, hey, make more. There's also what's called contact inhibition, where essentially cells that are touching each other will not actually divide. And contact inhibition is, of course, something that happens in animals, but it also happens in plants. You could do a little experiment where you have two of the same plant, and if you sort of touch one of the plants and let the other plant be fine, you will see a remarkable difference in the overall growth of that plant. Additionally, like, you'll see if you kind of grow a plant and it kind of becomes root bound, that is that structure, that process where they're kind of they're stuck, they can't grow anymore because they have this inhibition feedback. I guess I kind of went over a little bit of apoptosis ahead of time. Again, this is a programmed cell death. And again, there's a number of reasons why this may occur. Internal signals might be part of the overall plan, their body plan, or what's going on. A good example, oh, wow, this guy's good. Webbing between human digits, same sort of idea, where it is broken down. And then we can ask our buddies, the lysosomes, to come in and really break things apart when those cells are broken apart. And one of the good reasons for this is, of course, all that juicy stuff, all of the nucleotides in the DNA and all the material in the cytoplasm can very easily be reused within the body. And so it allows that to happen. So in terms of the cell cycle in cancer, what we're looking at is what happens when the cell cycle doesn't have the normal inhibition signals or normal check cycles to ensure that the cell cycle goes correctly and as planned and as it should. And essentially, cancer is a disease of the cell cycle where it recurs without end. So there's no contact inhibition. There's nothing like that. Instead, what you have is a situation where a cell is simply dividing out of control. And this process of carcinogenesis, where the development of cancer is a significant process that we've learned and continue to learn a substantial amount about. So aside from a cell that will divide uncontrollably, the result of that uncontrolled cell division might be a tumor or will be a tumor. Now, if this is a situation where, you just have a cell in one particular part of your body, it just loses all inhibitions, cycle, divide, and forms a tumor, and that is it. That is what is known as a benign tumor, but what we can also see is there are situations where instead of just simply creating a chunk of tumor, it creates cells that could then break off of the tumor and go elsewhere in the body, and even lead to other cells in other parts of the body having the same issue. So a situation where like a lung cancer might go elsewhere in the body and cause cancers elsewhere. And that is when you're looking at a malignant cancer. So cancer cells lack differentiation. They're not part of bodily function. But of course, that doesn't mean that they don't specialize. If you've ever heard someone talk about a tumor that had little teeth in it, it's not just some creepy sci-fi thing. It's actually something that can happen. If you think about it, all of our cells have the potential or have the genetic information for teeth. It's just fortunately, most of the time, our bodies do not express those genes. No one wants to have knee teeth or knuckle teeth or teeth or whatever. I am a watcher of horror movies, and I like certain types of horror movies, but when it comes to like body horror and that sort of thing, that really that's worth watching in my book. Anyway, when it comes to cancer, these cancer cells divide out, may in fact even be immortal. These may be cells that just divide and do not have any inhibition whatsoever. I mean, most cells eventually in their life sort of die out, and we'll talk about that. Perhaps you've read about telomeres as an interesting component of our understanding of the cell cycle. But it does not go through apoptosis. Tumors themselves are not going to be broken apart by any internal immune system response, because technically they're just really not good parts of you. The idea of metastases, the idea of something being a malignant situation where it travels elsewhere through the body to create new tumors elsewhere, of course, is a very difficult thing. As I said before, when it's benign, it's within a capsule. I guess as part of my sort of body horror, I do watch the occasional episode of our good friend Dr. Lee, who's the Dr. Pimple Popper. Don't know if you've ever seen that show, but a lot of times the lipomas and the other tumor-like materials, one of the things that she talks a lot about is that they have a capsule. And that's very important to have that capsule because it kind of maintains it so that it remains as a benign tumor in the body. This, in this case, being one usually of some sort of a skin cell or derm cell that's gone haywire. It's gone out of control. She's always concerned about getting the entirety of the capsule because if you leave some behind, it will then recreate the lipoma or recreate the various different skin conditions, which have caused emotional and personal anguish for the people who are coming to her, have them solved. So that is a situation where she is surgically removing the benign tumor, but want to ensure that she gets the entire capsule around it. Obviously, in situations where it's malignant, there is the possibility for invasiveness and may spread within the organ or within the body. Okay, is there a question? One of the students is requesting control. I'm okay with that, but I think that might have been an accident.
That was an accident, sorry.
No worries, no worries. Perhaps someone else wanted to talk about their experiences with Dr. Pimple Popper. Anyway, moving on. Moving on, in terms of the development of cancer, what we're really looking at when we're thinking about the cells and the DNA within the cells, you have a situation where there are often multiple mutations. There are what are called oncogenes or proto-oncogenes. An oncogene is a gene that essentially encodes for a cancer. And we all have different ones. We all have these in our bodies. And the proto-oncogenes are ones, are proteins that might actually lead to these sorts of problems. So we have suppressor genes within our genetic code, but it is when these proto-oncogenes mutate. A mutation within one of these essentially is the trigger that turns on the cell that makes it cancerous. And what essentially happens is there can also be mutation of the tumor suppressor genes, genes that encode for apoptosis that will kind of be able to help control cells that have not that have gone bad. So in this case, well, stop that. In this case, what we're looking at is a situation of epithelial cells. We're looking at a skin condition, so some kind of melanoma. So we will kind of get to that in just a second. So let's talk through that. We've got a situation where it's a melanoma, so you have a mutation, and essentially once you have a mutation and that cell divides out of control, it will spread that mutation, but then that would perhaps make it more likely those cells that are dividing so fast, essentially you're not going to have such a effective transcription, such a successful copying of the genetic code. And so the possibility, if it's going through this cycle a lot quicker, it also is more prone to mutations. So what happens is you eventually have multiple mutations within a bunch of cell materials. And within those that can kind of lead to the proto-oncogenes to become wholly active oncogenes, and essentially they could then enter into the body in a variety of ways, into the bloodstream, into the lymphatic stream. And when they get into those systems, when they bury from the initial melanoma into the muscle tissue and into ultimately the structures, the systems of the body that transport, that's going to be the highway to metastases and highway to all sorts of worse things happening within the body. And so essentially, this, I will say, use sunscreen, definitely try to take care of your skin. And if you have something concerning, please go to a dermatologist. They're kind people. I can recommend a couple. They're kind people. They're there to help. And it's very important. All right. So one of the things that happens with cancer that doesn't happen with regular cell cycles is there are structures called telomeres, and they are a certain length. And as a cell goes through the cell cycle, the telomeres are kind of lost, are kind of utilized. But what's unique about cancer is that these telomeres don't actually shorten, and that is a very significant thing, because at some point when the telomere has become too short, the cell can no longer go through the cell cycle. So this perpetuates this cycle in yet another way. Of course, there could be cell cycle genes associated with cancer, and of course, a variety of different other components of the DNA to structure. So there's a variety of different ways to do cancer treatments. Of course, you hear about radiation therapy, you hear about chemotherapy. These are aiming to make a tumor smaller or to stop the process of reproduction and to perhaps discourage metastases, as the case may go. But there are, of course, substantial side effects. My mother-in-law went through back in around November of last year, went through a process. She was diagnosed with stage 4 lung cancer, and she did 2 rounds of chemotherapy. After the second round, they determined that it was not effectively doing as much as they had hoped, and we arranged for hospice care, and within a couple of days she passed. So clearly she was very, it was a palliative sort of situation, trying to extend her life and trying to make her more comfortable. Honestly, at the end of the day, I'm not sure she was at a point where she should have received that treatment. But again, it's, you kind of have to have hope. And then unfortunately, that's, it's an unfortunate part of life. So, why is that? So a couple points to ponder. Why is apoptosis so important? Of course, it helps maintain and avoid cancer situations. It makes it so we're not all web-footed freaks. How does cancer develop in some cells more than others? Well, how they're exposed to mutagens. In the lungs, obviously, cigarette smoke is very, very difficult and very, very challenging to your single cells of the alveoli in the lungs. And so those cells can easily break down because of the mutagens in, say, a cigarette. And of course, something we're continually doing is trying to take our knowledge of the cell cycle to come up with new targets for cancer therapies. So Let's take a 5-minute break or a 10-minute break, and then we'll talk about meiosis, and there will be even less singing. So I'll see you soon. All right, so we're back, and we're going to talk about the second component of the cell cycle, which is meiosis. And what we're looking at, what we're thinking about with meiosis is, first of all, looking at how we can reduce the chromosome number. If we're all 46, how do we mate in the sexual process with someone else who has 46 chromosomes and still end up with an offspring with 46 chromosomes? So we're looking at a reduction in the chromosome number. The other thing we're looking at is shuffling of the chromosomes. While you do receive half your genetic material from your mother and half your genetic material from your father, the blend, that specific unique blend that is you, is really a component of which different chromosome numbers you get from which parents. So the first important concept here is the idea of homologous chromosomes, or homologs. Well, these are the pairs of chromosomes, because while we say we have 46 chromosomes, what we really have is 23 pair of chromosomes. And those pairs are matching or homologous. They're the same size, shape, same genes, the same material, the same information, or not information, but at least the same areas where those genes are found. And again, we do receive these each from our parents. So a child will receive one member of each of the 23 homologous pairs from each of their parents. And these pairs will contain, like I said, information about the same gene, and these are known as alleles. And we'll get into alleles again far deeper in the next section when we start talking about Gregor Mendel. Males and females both have 23 pairs of chromosomes, 23 pairs for a total of 46. And these chromosomes are known as diploid. This is 2N. Whereas the haploid number for the gametes in our bodies are in fact 23. So our regular cells, all of the cells in our body, have 46 chromosomes, except for the gametes within our body, which only have 23. And so that is where we get the haploid number, the 23, or the diploid number in most of our cells, which is 2N, or 46. Now, of those 23, 22 of those pairs, or 44 of those total chromosomes, are what are known as autosomes. These are things that will create, these are the things that will encode for what color eyes you have, some aspects of, say, your hair, or other components. And we'll talk all about those as well when we talk about the Mendelian side of genetics. We also then have the other two, the number 45 and 46 are our sex chromosomes, with the male being XX and, or sorry, the female being XX and the male being XY. And so this is a micrograph where they've actually managed to separate all of the, do a photograph of all of the chromosomes, and then look at those chromosomes and match them and pair them up. So this shows the 23. In this case, it is a male. So the sex chromosomes are not, in fact, identical because in this case, there's an X and a much smaller Y, but all of the other ones are paired. All the other 44 are paired. When they're like this, you can see that there are, again, just as in mitosis, two sister chromatids with a centromere in the center. So these sister chromatids are identical to each other. And then this chromosome has two sister chromatids identical to each other. One of those you got from mom, one of those you got from dad. In the case of males, you can see as it says here. So if we look at the life cycle, we have a situation where a fertilized zygote goes through mitosis 1 trillion plus times, going from a zygote, a diploid zygote, to a diploid baby boy, growing, or girl, or baby, growing through all sorts of mitosis to become fully formed adults. And then within the gonads of the male and female, where meiosis takes place, you have the shift from being all of these cells being diploid, all these cells being 2N, going through meiosis, and the female, of course, creating a haploid egg, the male creating the haploid sperm. When the haploid or 1N sperm fertilizes the 1N haploid egg, of course, you then go back from N plus N to 2N, and you have fertilization to form a zygote, and the cycle continues. So this is the life cycle that involves the cell cycle, if you will. Mitosis, as we said before, always important for kind of keeping and maintaining your tissues. The sole goal of meiosis in this situation, the two goals that we talked about, number one is the reduction in chromosomes so that you go from cells that are diploid to cells that are haploid, so that gametes have only one member of each homologous pair. So this is how meiosis looks. It starts with a single diploid cell and ends up with 4 genetically unique haploid cells. So no longer are we talking about situations where they're identical to each other and the parent. These two cells are very different from each other and different from the parent cell. In meiosis 1, you can see that it will go through the prophase, metaphase, anaphase, telophase, but in this case, on metaphase, the homologous pairs line up. It's called a synapsis, and you have basically these 4 sister chromatids all lined up, paired off with the centromere. So instead of having mitosis where they're all lined up in a single file, here you have them lined up with the homologous chromosomes. This set of larger ones, this set of shorter ones. So during the process of meiosis, you would then have the spindle will retract, and each of the daughter cells will have only 2 chromosomes, or the haploid number. Each will have 2 chromosomes, or only the haploid number. Now, we see a couple things happening here that are very important. So even though this individual has half of their genetic material from their mom and half from their dad, indicated by either red or blue, the setup or the structure of the tetrads are such that they will be kind of randomly, they will be random through on the metaphase, in metaphase along that equatorial plane. So what's interesting is that you can have a situation where because of where different chromosomes lie from this individual, from their parents, you can have a situation where basically a ton of one of your kids might like really look like their grandfather. And that is because of the situation. Essentially, what it's been said is that we don't really own our chromosomes. We're just borrowing them from our parents. So during meiosis 1, you have the tetrad, you have the homologous chromosome synapse along the metaphase. And in meiosis 2, the second cycle or the second component, you have situation where the individual haploid cells that have sister chromatids and a centromere will line up along the equatorial plate during metaphase, and they then divide. So in meiosis 2, essentially, these two cells are identical to each other and the parent. These two cells are identical to each other and the parent, but we have created new diversity based upon which one lined up where in this whole process. And again, this is just like the process that makes 4 sperm or a couple of eggs. And if you imagine the bajillions of sperm or the bajillions of possible combinations for an egg, you really are getting all the different diversity from that individual passed on to their offspring. So like in mitosis, meiosis 2, the sister chromatids are broken apart, like we see here. Now, I've already mentioned one area where meiosis really helps in doing, where meiosis really helps in different types of variation and variation within the species, where which lines up where is a very important component. Another thing that can happen when the tetrads are the homologous tetrads, the homologous chromosomes are all lined up, is that there is a situation where they can have what's called crossing over. So these homologous chromosomes synapse, and then what happens is when they synapse, there will be a switching of chunks. These chromosomes, these homologous chromosomes line up, and there can be exchange of material. And this is another way of variation to occur. So instead of just having the 2 blue and the 2 white, the 2 blue and the 2 red, you have situations where you have maybe one complete blue, one complete red, and then ones that are like 80% blue and a little red and vice versa. So now you have even more variation. So during prophase 1, the homologous chromosomes pair up, form the tetrad. We call this synapsis. Each tetrad has 2 homologous chromosomes, each containing 2 chromatids for a total of 4 chromatids. And the homologous chromosomes, the non-sister chromatids, will exchange genetic material, increasing variation. So this is the importance of this is, of course, number 1, maintaining the number of chromosomes within each new generation. And number 2, creating a variety of genetic variations. The possible combination of chromosomes in the daughter cells, the possibility of crossing over, and all the different possible combinations. That's just within one individual. And then when you take those variations and combine and lead to a fertilization with all of the combinations from the other individual, you have a situation where you really can have quite a bit of new variation within the species. So meiosis has two divisions of the nucleus, meiosis 1 and meiosis 2. And each one is broken down into 4 phases. The prophase 1 and 2, metaphase 1 and 2, anaphase 1 and 2, telophase 1 and 2. So in prophase 1, same sorts of things happen. You have the breakdown of the nuclear membrane. You have the chromatin constrict to form chromosomes. But instead of just having the individual chromosomes line up, you can see there's the synapses here where the homologous chromosomes will connect, maybe do a little crossing over. In metaphase, you can see the pairs lined up, and each of the daughter cells, the 2 daughter cells in this case, get a full set, a haploid number of chromosomes. They won't get a full pair of all of them. They won't be diploid anymore. This really is this reduction division where the number of chromosomes in the parent cell and the number of chromosomes in the offspring cell are different. And it has gone from being a diploid to a haploid. Still double-stranded chromosomes, which we'll deal with in meiosis 2. In meiosis 2, and prophase 2, the chromosomes condense, the spindle starts to form again, and then you have this lining up, not in homologous chromosomes because they've already gone through that process, but instead the haploid number of chromosomes lined up along the equator, along the spindle. The spindle retracts in anaphase 2, and ultimately you're left with 4 cells, each of which have a haploid number of single-stranded chromosomes. So in terms of comparing meiosis and mitosis, of course, with meiosis, there are 2 nuclear divisions, whereas mitosis only has a single one. Meiosis creates these 4 daughter cells following the final cytokinesis. Mitosis only creates 2 daughter cells. After meiosis, these are haploid and have half the number of chromosomes for that species, half the number as the parent cell of chromosomes. Diploid 2N parent cell, haploid 1N daughter cells. In mitosis, they are single-stranded, but they still have the same number of chromosomes. They still have the diploid number of chromosomes. They're identical to each other and to the parent. The only thing that this cell needs to go through in order to get these larger chromosomes is, of course, an S cycle. The only thing that needs to happen is it needs to grow. So what are some of the things that can potentially happen in chromosome number? There's what's called non-disjunction. This is a situation where the genetic material, in spite of the spindle's best effort, the genetic material is not divided evenly. Instead, the synapse tetrad end up going, for one particular chromosome or another, end up going into a single cell, and the other cell doesn't get any. Another situation that can be considered non-disjunction would be in meiosis 2, when the sister chromatids don't separate. Again, the spindle work of that particular cell process not happening effectively. So one example of this non-disjunction, which occurs during meiosis 1, is trisomy 21, where there are 3 copies of chromosome 21. So you can imagine what happens is during meiosis 1, the chromosome 21 pair will line up, but when anaphase 1 occurs, instead of each side or each cell getting a single chromosome, one cell gets none, the other gets 2. And so all the other chromosome pairs, all the other tetrads, all the other synapsed chromosomes will divide evenly. And so in that process, that cell is still viable that has the extra chromosome 21. That cell is viable, and when it is fertilized, you end up with an individual with Down syndrome. You end up with an individual that has actually 47 chromosomes. All the pairs plus the extra chromosome 21. Another example, the opposite of that non-disjunction would be a monosomy, where you have a single copy of a chromosome. You don't actually have the second one because of that. Now, there are so many different variations, and in general, we try to present a simplified version of things when it comes to things like biological sex, but there are truthfully a lot of variations. A good example of monosomy would be a woman who only receives one sex chromosome. They only receive one X chromosome. So they are known as X0 because they don't have 46 chromosomes, they have 45. And they are still female, but they don't necessarily have all the female characteristics, and they are not fertile. Another example would be an example of a trisomy when it comes to sex would be Klinefelters, where an individual receives 3 sex chromosomes instead of just 2. They would receive basically 2 XX from the mother and then a Y from the father. And so they'd be XXY. And again, they have a Y chromosome, but they have the appearance, or they develop into a female, one that one that isn't fertile as well. You could even have a situation where an individual is XYY, where they actually have a trisomy in the sex chromosomes of XYY, and they are basically, they have twice the amount of male. There was actually a study done once that actually determined whether the population within the prison had more XYY men than the general population in terms of percentages. And it turns out they did not. They thought maybe sort of the extra Y chromosome would make them extra testosterone, extra aggressive, what have you. Now, again, this is just looking at this situation from the chromosomal component, where there is the potential for variation. Of course, as you get more into understanding development, you can have situations where individuals might be XX, but they may have what's called chorionic hydroplasia and essentially will appear female, or excuse me, will appear male, even though they have XY. And there might be someone who is XY who has the inability to identify testosterone. And because testosterone and estrogen are so closely related, it's important to kind of think about all of these shifts and changes. And the important thing here, again, is not to necessarily try to, scientifically ascribe or do analyses on people on what they are. The whole idea of gender is, of course, more of a social construct. We're just simply looking at at sex and chromosomes. But the fact of the matter is that if you look at the types of variation that can occur, even at the chromosomal level, and get into all the other stuff, I think our understanding of gender is really as a component of biological sex is very interesting, and I think it is worth sort of an open mind when it comes to these sorts of things. So that's my little 2 cents on it in terms of trying to understand it from a biological perspective. What I'm essentially saying is sort of the XXXY, when we talk about sex chromosomes, really are kind of are a massive simplification of things, because really biology has so much more nuance and so much more interesting things going on beyond the chromosomes and then the genes and how they're expressed. So I find that a lot of people that perhaps only really paid attention in like 7th grade, where XX and XY is all there is, might perhaps not have the full education, and their simplistic understanding of science perhaps has led them to make certain assumptions about the world around them or whatever. That's enough of that. Okay, so again, what actually happens with an individual with trisomy 21, the pair of homologous chromosomes during the process of meiosis? One, there is not the even splitting where each of the daughter cells gets one of the chromosomes, and the gametes will have one less. These gametes will have one less, and these gametes will have one extra chromosome. In term, of course, it's quite common. It's called not, quite common, relatively speaking, in terms of the known non-disjunctions. I think perhaps some of the reasons why it is so known is because, of course, there are a number of recognizable characteristics, various different physical features that allow someone who has Down syndrome to be identified pretty easily as having that, beyond just simply the mental or internal disabilities. I work with a number of I work at a cafe that does job training for those with various different physical and intellectual disabilities, and that's sort of one of my 18 jobs. And what I working with them in a cafe setting where they can get job training, where they can learn how to interact. I work with a couple of kids always with making change and kind of helping them with that component so that they could potentially enter the workforce and be a little bit more sustaining and, essentially less, not a burden, but less of a cost to the government. You always want to have that safety net for them, but this is very important, and work with a number of individuals with Down syndrome. And they're great to work with. They really are. Yeah, I'm just gonna no. This just no. This is not right. Okay, we're just gonna do something about this. Apoptosis, no. All right. All right, we need to fix this. Um All right, we're going to cross that. Okay. More recent research has indicated that this particular understanding of trisomy is a bucket of crap, and some people think it's sort of like pushing women of a certain age to worry about biological clocks and this and that sort of thing. But really, more recent research about this and more recent actual birth rates have indicated that this is not the case. So I leave it in there so I can make a big show out of crossing it out. Anyway, all right. So here is a young boy with Down syndrome, and you can see in this case, it's a male, but in this case, when you look at the homologous chromosomes on this what's called a karyotype, this image of all the chromosomes and pair it up. Essentially, what you have to do is you take a picture of it, and then you have to cut the little cut the photographs of the chromosomes apart. There's really no way to get them to synapse this way. But here you can see, in this case, there was a non-disjunction, and this young lad got an extra chromosome number 21.